This is a great time to be a human. Homo sapiens are experiencing a population explosion that is exponentially increasing the amount of individual organisms of our species all over the planet. Put simply, we can mold the world now to suit our specific biological needs. That's what humanity does. Yet in making our environment more suitable for our survival, other species, a lot of other species, are becoming extinct, disappearing in greater numbers as our impact on the Earth grows. Humans are affecting the despecification of our planet. Humans need healthy ecosystems to essentially exploit for our own survival. And the key to a healthy ecosystem is genetic diversity. Genetic diversity leads to biodiversity, and biodiversity creates ecosystems that offer products of nature that are necessary for our survival. So, in the next few minutes, we'll define genetic diversity, how we identify it, and how we're losing it. We'll discuss species-specific population and how those species are affected by the size of their gene pool. And finally, we'll talk about what modern conservation geneticists are doing to maintain genetic diversity and how they can now restore it in populations that are on the brink. But in order to continue this conversation, I think we should go someplace else. In terms of conservation genetics, we can talk about genetic diversity in two main ways: interspecific and intraspecific. Interspecific diversity in an ecosystem refers directly to the genetic differences from the very smallest organisms to the very largest organisms. Intraspecific diversity refers to the genetic differences in each individual species. To go slightly deeper, genetic differences inside a species are analyzed as intrapopulation diversity, or the genetic variation occurring in one population of a species, and interpopulation diversity, or the genetic variation among different populations of a species. Scientists no longer look at phenotype only, as Mendel did, to identify genetic diversity in a species. Currently, geneticists take whatever samples of DNA they can find, remains, plant matter, fecal matter, seed, or direct cell samples, then use polymerase chain reaction to generate and analyze the short tandem repeat sections of the sample genome. In this way, and others, conservationists can work towards certain goals. They can see how and where species are migrating over generations, they can identify the level of interpopulation diversity, and they can ultimately assess the viability of the threatened species. When specific populations of a species become isolated or endangered, their unique alleles may be cut off from the rest of the population. Unable to share that genetic variation with the greater population may eventually cause major problematic effects. General knowledge tells us that inbreeding, or breeding among species or populations with reduced gene flow, can be highly problematic. Populations with that reduced gene flow or decreased genetic diversity that do end up breeding increase their chances for genetic phenomena, and that in turn increases their chances of extinction. Scientists have determined that populations of 100,000 individuals show long-term sustainability. Populations of 10,000 individuals are likely to be limited in adaptive genetic variation. Populations of 100 individuals are extremely sensitive to problems, and populations of 50 or less are likely to become extinct within 50 years. When a population bottleneck or a one-time massive decrease in breeding individuals occurs, the effect can be seen for generations. A population derived from a small subset of individuals that has lost genetic diversity exhibits difficulties in long-term survival. This is known as the founder effect, and these populations require the reintroduction of diverse alleles. So how do conservationists reintroduce diverse alleles into populations? Well, there are two ways. One is called in situ conservation, and one is called ex situ conservation. In situ conservation, scientists make a concerted effort to protect the number of organisms that currently exist in that ecosystem. This must happen before a certain tipping point where the loss of genetic diversity becomes too much. After that, in situ conservation can still be done. This happens when scientists transplant members of one isolated population into another isolated population. The attempt here is to recreate that genetic diversity by exchanging genetic material through different populations. If these both fail, ex situ conservation is necessary. This is performed by transplanting wild organisms into a zoo or a lab where a structured captive breeding program is established. Ex situ conservation can also be done by utilizing gene banks. In these gene banks, samples of genetic material are taken and cryopreserved for times when those genes will be necessary. Previously harvested genes can be added to threatened populations, effectively reintroducing genetic diversity without the need for natural processes. Gene banks are essentially the next era of genetic conservation. Thank you for spending time with me today to talk about conservation genetics. You know, in the end, the Earth will have another mass extinction event, and everything that's pretty much here will be gone. But that's really not the point. The point is, as humanity, we should respect the things that are around us and enjoy this time that we have to the best of our ability. Take note of what we talked about today. I hope you learned something, and I hope you can pass that on.